Tinakoto, and welcome to what I hope will become a regular Bioc 351 feature, a biochemistry highlight for the day. All going well, I'll post the highlight next on Monday the 6th. This material is 100% non-examinable. But first, thanks for your enthusiastic participation in our lab Zooms this week. You made it very successful. Stay in touch with us and stay in touch with one another. Today's highlight is one I learned about in the International Union of Crystallography journal, a publication even I find a little bit dry usually. But let me share it. This article grabbed my attention by explaining that in the 1950s, X-ray crystallography was very au courant, even mentioned in popular movie dialogue, much as we might mention AI or quantum computing in movies today. This article's URL should be visible at the top. However, it wasn't this article itself which got my attention. This article pointed me to this URL on how X-ray diffraction maps and atomic structures inspired graphic design at the Festival of Britain in 1953. The resulting designs were used on china, on neckties, fancy lace, floor coverings, and even ashtrays. Visit the URL at the top or just Google Festival of Britain Pattern Group to see some of the things they made. One of the designs was based on Dorothy Hodgkin's Patterson Map of insulin. This is the Nobel Laureate Dorothy Hodgkin, and some of the information about her getting the Nobel Prize for insulin. That Patterson map is shown on the virtual diagram behind me here. Now, a Patterson map is related to an electron density map, but different in that it can be drawn before you know the phases that you need to draw an electron density map. It shows not exactly where the atoms are, but it shows the relationships between them. By analogy, if one were to draw a map, an electron density map of the customers waiting to get into New World, one would have a series of peaks spaced about 2 meters apart, but with some variation, some 1.8, some 2.5 meters apart. The Patterson map of this would also show the 2 meter spacing between objects, but would show an average and would not show where each customer is in the New World Mall. Rather, it would show where, or show where each peak is in the unit cell. The Patterson map is more complex if the line turns a corner and you introduce a second dimension. For instance, the 6-fold pattern that shows up in the middle here, that 6-fold pattern there, is the result from just 3 atoms in an equilateral triangle. So how does that work? Depending on which of the 3 atoms you take as the reference, you can find another 2 atoms at 1 o'clock and 3 o'clock, or take the apex of the triangle at 5 o'clock and 7 o'clock, or 9 o'clock and 11 o'clock. This map here was the one drawn and colored by Dorothy Hodgkin herself for her lab notebook as she worked through the phase problem to solve the 3-dimensional structure of insulin. In your lab notebooks, strive to make both the substance and appearance of your notebook as timeless as Dorothy Hodgkin's notebooks. Speak to you next week, and until then, be well.